belongs from the middle of my sentence. That's okay. It's lovely that we've all come together to listen to Dr. Rony Khnayzer, Associate Professor of Chemistry, but also Director of Liberal Education in the School of Arts and Sciences. And I'm fascinated to hear what you have to say about photochemotherapy. Blue light at the end of the tunnel is the title of your talk. So once you're done with your speech, we will take Q&A. But in the meantime, if any attendee has a burning question they want to ask and they want to record it in the chat, you can feel free to do that. And then we will invite you to contribute to the Q&A at the end. So without further ado, I'm going to hand over to Dr. Khnayzer. Looking forward to it.
Thank you very much, Dean Jnaynati, for the kind introduction. I'm gonna share my screen. All right, so in today's talk, I will be telling you a story that we have developed in the lab at LAU about photochemotherapy. More specifically, the use of blue light in chemotherapy. Now, why I'm interested in research on cancer? Well, back in 2013, when I joined the faculty at LAU, I was a photochemist by training, more specifically looking into inorganic molecules and chromophores that will harvest the energy from sunlight and store it in the form of hydrogen. So my research was about solar energy conversion schemes, as well as sensors applications. If I want to realize and continue my research at LAU, we needed multi-million-dollar equipment. So I looked around me. Those equipment are not present, but I had wonderful colleagues in the School of Arts and Science, and specifically in the Department of Natural Sciences, who are doing excellent work in cancer research. So I thought how I can contribute there in my with my expertise in designing chromophores. And this is how the journey started. Now, cancer by itself is not a single disease, and it's a global problem. The report estimates that in 2020, there was 1.8 million new cancer cases and 600,000 cancer deaths in the US alone. And you can extrapolate to the entire world from these statistics. What is interesting to look at are the fact that cancer incident rate did not decrease significantly over the past decade, while cancer death rate declined by only 1.5% annually due to advancements in therapeutics as well as healthcare systems. Now, how an inorganic chemist can contribute in the field of cancer? When we did the literature search, and I had to learn a lot during my journey, and I'm still learning. I learned from my students, I learned from my collaborators, and of course, from the wonderful postdocs that are hired at LAU. We can see that cisplatin, which is an inorganic platinum complex, a square planar simple complex, is one of the widely used and highly effective chemotherapeutic agent used for numerous types of cancer. So this is encouraging for me. I can find some kind of mimics to cisplatin and overcome the limitations that cisplatin has. Among the limitations that are highlighted in the literature are adverse side effects, and we can see that later on in the data we have acquired on cisplatin controls, toxicity, narrow spectrum of activity, along with a very important notion of intrinsic or acquired resistance by cancer cells, partly due to the open coordination sphere that platinum has and the square planar geometry that characterizes platinum-2 complexes, so allowing nucleophilic attack of sulfur-containing compounds and other nucleophiles into the axial position of the platinum center. Now, the development of cisplatin, as well as its successful use as anti-cancer agent, inspired research on metal-based anti-cancer drugs. In this talk, I will focus on our efforts in the field of photodynamic therapy or photochemotherapy, and specifically, I will talk about advances in science while using copper and ruthenium complexes. Now, what is oncological phototherapy? It relies on administering some kind of light-absorbing molecule or chromophore. Then the chromophore is distributed everywhere in the body. Then using spatial and temporal control of light delivery, we can selectively trigger a photochemical or a photophysical process or transformation that are useful for chemotherapeutic applications. One of the most widely used chemotherapeutic agent that is FDA approved is photofrin, that is specifically useful in esophageal and non-small lung cancers. It is based on a porphyrin molecule that is capable of absorbing light and converting it into a useful form of energy that can trigger oxidative stress, the formation of reactive oxygen species, and lead to cancer cell death. Now, encouraged by all of this literature, we focused our studies on 3 different mechanisms and chromophore employment in those mechanisms. The first one is photodynamic therapy. It involves the administration of photosensitizing agent. So when these photosensitizing agent absorb the energy of light, they produce a triplet excited state where the electrons in the excited state are basically parallel in spin. This triplet excited state, for my dear chemistry students, this is a long-lived excited state, and due to its long lifetime, it can undergo energy and electron transfer processes in a type 1 and type 2 reactions, leading to oxidative stress, cellular damages, and ultimately to cell death. However, the major drawback of photodynamic therapy and the employment of sensitizers in this scheme is that they rely heavily on the presence of oxygen. To that end, and to solve this issue, photoactivated chemotherapy relies on caged metal complexes or other type of organic complexes, but here I'm specifically talking about the use of inorganic molecules in this scheme. So I term that metal ligand complex. This is a caged complex, a prodrug, an inactive molecule that is administered to ideally to patients, and then upon controlled visible light irradiation, we can split the molecule into the free metal that can coordinate to water molecules and the free ligand. And then either the metal or the ligand will bind to DNA or protein targets, inducing cell death. Now, in this scheme, there is no oxygen that is needed, and it will work very well in the hypoxic environment of tumor tissues. However, in the literature, we found that the role of each entity that is produced following photochemical reaction is ill-defined, and this is addressed in part 2 of my talk, where we highlighted the importance of the contribution of ligands in certain cases, as well as metals in other cases in photochemotherapy. Now, the third type of therapy is to directly use the energy of light to trigger certain kind of biological or physiological effects. Now, the phototherapy concept dates back to centuries ago, when exposure to sunlight was used to treat skin conditions. And it's still being used in certain cases of psoriasis, atopic dermatitis, and jaundice in young kids that are born with this condition, and blue light is used in the form of LEDs and exposing the body to this radiation can help mitigating these conditions, these adverse health conditions. Back in 1903, Finsen won a Nobel Prize when he discovered the efficacy of light in treating lupus vulgaris. And this is due to the bactericidal effects that were discovered of light, as well as the stimulating effects that light by itself has on tissues. Now, if we're wondering how light can penetrate tissues, it really depends on the wavelength of light, with UV light the least penetrative, and as we go more and more in the visible, we get more and more penetration into the tissues, up to around 900 nanometers, so in the infrared, where we start losing the penetration effect due to intrinsic absorption of these wavelengths by water molecules. Now, how light by itself can trigger photobiological response? Well, there should be intrinsic molecular chromophores that exist in our body. So when we dig in the literature, we found out that there is a wide range of metal complexes that exist in our body, and more specifically, a distinct porphyrin-based molecule that exists in cytochrome C oxidase unit that is involved in the electron transfer chain. So upon light absorption, what happens is that there is a stimulation of the mitochondrial electron transfer chain that will lead to some leaked electron through the redox processes, and these leaked electrons will help in the formation of reactive oxygen species, and ultimately leading to the physiological function of pure light. So here in this slide, I show you the structure of the cytochrome C oxidase, and more specifically, the structure of the chromophore that is built within this moiety. This is the UV visible spectrum that shows the absorption of light by this natural chromophore. We can see that at the blue region of the UV visible spectrum, around 450 to 460 nanometer peak, we have a Soré band, which is attributed to the pi to pi star transition of the porphyrin ring. So this is a distinct high energy absorption band in the metal complex that will help us in basically the light absorption and modulation of the biological activity. So in the first part of my talk, I will focus on the use of copper sensitizers in photodynamic therapy. Why copper? Because it's earth abundant. It's a mineral that is present in the body. It has tunable photophysics, and this is exactly why I am interested in this, because as a chemist, we can tune the activity in a biological environment by tweaking the structure of the photosensitizers. It has a specific tetrahedral geometry as opposed to the square planar platinum geometry. These copper complexes do not undergo side reactions by and deactivation like their cisplatin analogs, and they have been found to have anti-cancer activities both in the +1 and +2 oxidation state. However, to the best of our knowledge, there was no copper-1 complex that has any useful property reported in the literature as photodynamic therapy agent. And this has a reason. When we excite copper-1 complexes from the ground state to the excited state, there is a distortion of the tetrahedral geometry back to the square planar geometry. This is coined as pseudo-Jahn-Teller distortion. And this flattening of the molecule allows ligands such as water and biological medium to transiently bind to the excited state of the complex, deactivating the excited state. And if you remember when I showed you the scheme for photodynamic therapy, we actually need to harvest the excited state properties. So when I thought about this problem, we basically came up with a very important direction if we want to develop the first ever copper-1 photosensitizer, and specifically copper-1 bisphenolphthalein photosensitizer for use in photodynamic therapy. First, we need the molecules to be absorbing visible light. So this is easy. We can lock the geometry and tune the photophysical properties. Second, we need the molecule to be stable in the dark, stable upon light activation, and we need to lock the geometry so that it doesn't undergo this Young-Teller distortion in the excited state. And this can be done through including sterically bulk ligands on the 2,9 positions of this phenolphthalein ligands. So the molecule, when it goes to the excited state, it cannot undergo flattening distortion due to this steric strain. Of course, all of these molecules must be water soluble. That's why a brilliant undergrad in my lab, he developed a technique, a novel technique to make a highly pure copper-1 complexes in the chloride form through counter-ion metathesis. It's a bit beyond the scope of my talk today, but I just wanted to highlight the contribution in that field as well. So if we take a look at the biological activity of prepared copper-1 complexes, bisphenolphthalein complexes, we can see that as we increase the steric bulk from methyl to phenyl to sec-butyl groups, what we have typically, we have an increase in the steric lock. And this was actually nicely correlated with the biological toxicity of these metal complexes that are tested on cancer cell lines. So briefly, these figures here, they show us on the y-axis the percent viability of cultured cells in vitro, and on the x-axis, the log of the concentration of the drug that was incubated with these cancer cells, both in black under dark conditions and in red upon light activation. We can see that at low concentrations, we have close to 100% survival because the molecule needs a certain threshold concentration to start killing cancer cells. And then as we increase the concentration, we have a drop in the viability or increase in potency or cytotoxicity. IC50, or the concentration at which 50% of the cells are still alive or 50% death, is a measure for us to look into the potency of the drugs. So the lower the IC50, the higher is the potency of the drug. We can clearly see that for all of these molecules, we have IC50 or cytotoxicity that is almost equal between dark and light, meaning that those molecules are not useful for the photodynamic therapy of cancer. However, upon the introduction of disec-butyl groups on the 2,9 position, we found a huge increase in cytotoxicity upon light exposure with a decrease of 10 to 80 folds of IC50 depending on the tested cell line. So we have reproduced these results on 2 cell lines, and then we have moved into a thorough mechanistic study in order to elucidate how this copper-1 complex is capable of catalyzing the photodynamic therapy of cancer cells. So what we have found is a clean and a clear increase in the phototoxicity index. This index is simply, again, showing you how good a photosensitizer is in phototherapy. So it's the IC50 in the dark over IC50 up in light exposure. The higher this number, the more is the change in potency up in light exposure. We see that only the molecule that has the microsecond lifetime exhibits basically a phototoxicity index that is significant. And subsequently, we have developed the first reported copper-1 bisphenethylene complex that is capable of being used in photodynamic therapy scheme. Interestingly, this copper-1 complex, as expected, has some biological target, and the target was DNA. So we can clearly see using UV-Vis experiments that the molecule binds to DNA and changes its photophysical properties accordingly. Up in fluorescence microscopy experiment, we see clearly the intrinsic emission of this molecule, because now the molecule has a high lifetime in the excited state, so we can see the emission of light emanating from these molecules inside biological targets. We can colocalize the drug with the nucleus through a colocalization study with nuclear stain, such as DAPI. We have found that also the molecule has slight diffuse cytosolic presence, in addition to a large concentration in the nucleus. We also found that the molecule, upon light irradiation, increases the production of reactive oxygen species, which is a key step in catalyzing the cancer cell death. In addition, using biological experiments, our collaborators in the biology lab found that the molecule induces severe DNA damages that are pronounced relative to the positive controls in these experiments. So in brief, what we have discovered in the first part of our journey was the use of copper-1 bisphenethylene complexes for photodynamic therapy. We have elucidated that these molecules, they interact with DNA, and we have shown that the control of photophysical properties through a fine-tuning of the structural lock in copper-1 sensitizers is essential to increase their use in these photodynamic therapy applications. More detailed mechanistic studies are underway, including cellular uptake and in vivo experiments on mice. Now, in the second part of my talk, I will switch gears and tell you about how we can develop chromophores that are useful for photoactivatable chemotherapy. So in that particular part of my talk, I will focus on strained ruthenium complexes that are capable of producing drugs upon light activation. Why ruthenium? Because again, we have a handle on the photophysics and photochemistry through ligand modifications. It offers us the potential of producing octahedral ligand framework rather than the square planar framework of cisplatin, and this is advantageous when it comes to decreasing cell resistance. It was shown that ruthenium-2 and ruthenium-3, they have anti-cancer, anti-metastatic, and effects in vivo and in vitro. And in some studies, certain ruthenium complexes were found to selectively accumulate in cancer cells. So the selective uptake or the selective targeting of cancer cells or cancer tissues is of a great use when it comes to therapies and therapeutic potential of drugs. Now, what is the design strategy? I told you in copper complexes, we had to lock the geometry and we had to keep the molecule stable. In ruthenium, if we want to make use of the energy of light to destroy the prodrug and form the drug inside the cells, we need to actually go against stabilizing the complex and destabilize these complexes. Now, typically, when we excite ruthenium chromophore to the excited state, we also form the triplet state. However, we have a dissociative metal-centered state called DD state. I'm not going to go into details here, but these states can be thermally accessible from the metal to ligand charge transfer excited state. Remember, in copper, we had this state with a long lifetime. It's emissive. In the ruthenium, if we have a perfect octahedral geometry, this excited state is great. We can harvest it. We can use it in photodynamic therapy. However, if we do slight ligand steric strain, what happens is that the ligand field splitting will decrease, lowering the dissociative triplet DD state or metal center state, decreasing the activation energy to reach this dissociative state, and subsequently allowing more and more photochemistry to occur, while prodrugs can be readily converted to active drugs. Now, this photochemistry was used to our advantage in photoactivatable chemotherapy schemes, and we have developed a series of ruthenium bis-bipyridine. So this is the bipyridine ligand. So we have 2 bipyridine on all of these complexes with systematically varied third ligand. These are bidentate ligands that bind with coordination to the ruthenium metal center through a coordinate bond with nitrogen. So this is a pure phenanthroline. Then we have dimethylphenanthroline, we have diphenylphenanthroline, and we have a prototypical dimethyl bipyridine, which was previously reported in the literature for comparative purposes. Interestingly, we have measured the crystal structure. We have elucidated the crystal structure of these compounds, and we found that this molecule, as expected, exhibits perfect octahedral geometry, but a huge distortion from octahedral geometry was achieved through the incorporation of these sterically bulk or hindering groups on the 2,9 positions of phenanthroline. When we went from the methyl to the phenyl groups, the distortion was very pronounced, and we could find that the dihedral angle between the ruthenium nitrogen plane, which is depicted here in yellow, and the plane of the ligand, instead of being 0 in a perfect octahedral geometry, is now up to 20 degrees. And this distortion was consistent with one of the reported literature that shows a correlation between other types of complexes and the dihedral angles. Interestingly, and this is for the first time, we show that we have intramolecular pi-pi stacking between the phenyl arms that are on one of the ligand and the pyridine rings on the other ligand. This intramolecular pi-pi stacking will have a huge implication on the photochemical properties of this complex. So in NMR spectroscopy, we found evidence that of this intramolecular pi-pi stacking in solution. The evidence for the crystal from the crystal structure was clear and tabulated. A broadening of the proton NMR peaks of the phenyl groups were evidenced in NMR spectroscopy. And up in light irradiation, and this is the interesting part here, when we shined light on this complex, we exclusively ejected the non-sterically straining ligand, which is unusual in photochemistry, but it is expected here since this molecule is held together on the ruthenium center through the arms that are undergoing intramolecular pi-pi stacking. Of course, besides the fact that bipyridine ligands are by nature more photochemically labile due to free rotation around the C2 prime bond. Now, once we characterized the photochemistry and we correlated the crystal structure parameters and the distortion in the crystal structure with the photochemical properties, we have moved into looking into the excited state dynamics. So when we shine light on these molecules, we can probe using ultra-fast transient absorption spectroscopy the excited state lifetime, and we see a clear and nice trend as we go more and more towards more sterically hindered structures. We have less and less excited state lifetime. So here, excited state lifetime works to our benefit when we reduce it, since we are populating on the other side dissociative states and we are undergoing photochemistry. So in this part of the project, our aim is to reduce the excited state lifetime and populate dissociative state instead. We have basically also correlated the excited state lifetime with the activation energy through Arrhenius plots. So these are temperature dependent emission plots. We are reproducing these results currently in the lab, and we are expanding the knowledge about the photochemistry of these molecules as well. Now, what is our contribution to the field? Well, we have highlighted the importance of the photochemical products, the various photochemical products in the cytotoxicity of these complexes. This was ill-defined in the literature, and our contribution was highlighted on the cover of Dalton Transactions, which is a leading journal in inorganic chemistry. Not only we have developed the knowledge on what contributes to cytotoxicity and how, we have also developed a new technique based on liquid chromatography mass spectrometry to characterize these molecules in biological media, namely in urine and blood, using US FDA guidelines. And also our contribution to this field was highlighted on the cover of Analytical Methods journal, which is published by the Royal Society of Chemistry. Importantly, when we shine light on these molecules, we see a mixture of ligands being ejected, as well as a mixture of metal complexes, in the exception of one of these complexes, which bear the diphenylphenanthrene ligand framework. So when we put these molecules into test and we do the appropriate controls, we see that the ligands by itself that are ejected, specifically the dimethylphenanthrene ligand, exhibit a huge cytotoxic effect on cancer cells versus the ruthenium bipyridine bis agua complex, which was thought in the literature to be the leading cause of death in photochemotherapy schemes. We found that this molecule is actually inactive when tested on various cell lines. And if we digged into the literature to try to support our results, since it's a controversial results, and we found out a Nature paper in 1983 that looked into the intrinsic toxicity or basically activity of phenanthrene ligands in the presence of endogenous and added copper metals, and it shows that these ligands are very active and decrease the growth of cells in vitro, even though their aim was not really cancer or anything. But we have found evidence in the literature that our results are solid and they are valid and constitute an important parameter to be looked at every time we want to characterize these photoactivatable chemotherapeutic drugs in vitro or in vivo. Now, on the other side, when we have seen the photochemistry of various ruthenium complexes, we found out that this champion ruthenium molecule undergoes a very distinct photochemistry due to the intramolecular pi-pi stacking. We have a clean ejection of, I call it an innocent ligand, because this ligand is not biologically active, while it leads to the formation of a unique ruthenium agua complex. And why I'm saying unique, because the only way to synthesize this complex is through photochemical processes. We found that this ruthenium-2 molecule is undergoing cyclomethylation to the ortho position of one of the phenyl rings with a single agua complex that is bound to ruthenium. And we have now a study to purify this product and characterize it in different media. We are very optimistic about the use of this molecule that is synthesized photochemically for pure chemotherapeutic applications. Why I'm saying I'm optimistic? Because our drug is very active in the presence of light, but not in the absence of light. So this really acts as a nice prodrug for photoactivatable chemotherapy approaches. We see that the ligand has minimal activity and only is active at high concentrations. We see that the perfect octahedral geometry of the ruthenium analog of the strained molecule, as expected, shows no significant change in cytotoxicity upon light exposure, further verifying that our photoactivatable chemotherapy schemes are important and key to the success of this type of application. And of course, when we do a parallel measurement with the prototypical cisplatin, we see that the photoproduct has much more potency on an array of cell lines relative to cisplatin. Now, interestingly, when we looked into these photoproducts on cancer cells versus normal cells, mesenchymal stem cells isolated from the bone marrow of rats, we found that the molecules are much more potent on cancer cells versus normal cells, whereas the uptake of these complexes, as measured by ICP-MS and LC-MS measurements, was almost the same and did not significantly change between cancerous versus normal cells. This triggered us to think or to hypothesize that these molecules, they have intrinsic selectivity, or at least partial selectivity when it comes to interaction with cancer cells versus normal cells. And this also encouraged us to go beyond in vitro characterization and go to animal models to characterize the activity of these drugs. To that end, an in vivo model based on chemically inducing tumors or carcinogenesis model was performed. We used the NBA TPA model to grow tumors or basically cancerous or precancerous tissues in mice. And then we followed the growth of these tumors for up to 8 weeks. As expected, for the controls that were receiving only saline injections, the tumor kept growing as a function of the weeks. So here we have the tumor volume, and here we have the weeks of therapy. So this is our control, which expectedly increased in terms of total tumor volume, whereas mice group that were exposed to our photoactivated drug, and these were done through intra-lesion injection of the pre-activated drug under the skin, we saw a significant decrease in tumor volume and tumor incidence after 8 weeks of treatment. Importantly, we saw no change in the physiological functions that were tested, or any decrease in weight of the mice in the treated group or any obvious change in behavior or serum biochemistry. Even the mice could sustain up to 100 milligram per kilogram body weight of injection of these drugs without any observed death in the treated group, reflecting some kind of selectivity of the drug on cancer tissues. Now, as a conclusion for this part of my talk, which is a summary of years of research and publications in that area, we have synthesized and characterized a series of ruthenium-2 polypyridyl complexes showing interesting photochemical behavior that is correlated with the structure of these molecules. The mechanism of cancer cell death was likely due to ligand dissociation and the formation of cytotoxic ligands in certain cases. And this part was very important in the literature to highlight the role of ligands in mediating cytotoxicity. And there was another report that came after our publication that also verified our results and hypotheses. And in other cases, we have shown that the metal agua complexes that are formed post-irradiation are extremely cytotoxic and maybe selective against cancer cells. We're still looking into the mechanism of action of these drugs in vitro and in vivo. Now, the last part is just focused on a serendipitous discovery of the role of pure light irradiation on reducing tumor volume and tumor incidence in superficial tumor models in vivo. And that's why I entitled my talk, Light at the End of the Tunnel. Now, is light really effective against cancer? The answer in the literature is not very elucidated. You can find here and there certain reports that light indeed decreases the viability of cancer cells, especially the blue light. In some cases, they did experiments on colon cancer cells for days of exposure to low intensity blue light, and they showed some apoptotic potential and anti-proliferation of cancer cells. In addition, blue light was also shown to exhibit effect on B16 cell lines, and exposure to blue light also inhibited migration and invasion, which are important anti-metastatic effect of light, both in vitro and in vivo. But to the best of our knowledge, there is no reported data in the literature that shows a clear therapeutic modality for the use of light for tumor regression and for the decrease in tumor growth. To that end, we have used three types of wavelengths of light, one in the red, another in the green, and third one in the blue and the white spectrum composed of RGB or red, green, and blue. We find that the red and the green wavelengths did not significantly decrease cell survival up to 9 hours of irradiation. Of course, here we're looking at milliwatt per cm squared irradiation, so it's not extremely intense light. Whereas the blue light or the white light composed of red, green, and blue decreased significantly cell survival, allowing us to look into the phototherapy potential of light by itself without the use of any chromophores or any inorganic complexes for cancer therapy. In a similar model, and here we show again the controls where the tumors are growing in the presence of the negative control or mice subjected to simple saline injection. Here we see the cisplatin group. The group of cisplatin suffered from severe toxicity, since cisplatin is not very well tolerated in this model and does not have a huge significant change in tumor volume and tumor incidence, whereas the group that was subjected to pure light irradiation, so here's the setup. You see that the mice are lined up. This is the tail of the mice, and we have a controlled irradiation through a high power white light. We do this irradiation thrice weekly, so 3 times per week for a period of 90 minutes each. So it's really a treatment modality. So we bring in the mice, we treat them with light, and then we put them back in their dark cages. We see a huge decrease in the tumor volume and tumor incidence in the group that was exposed purely to light irradiation. This was coupled with a nice results that these mice did not exhibit any side toxicity upon light exposure. Even we didn't see any skin irritation. These mice were controlled in terms of temperature exposure, since we have irradiation with a water filter. This filters out any potential production of heat in a confined environment. And we didn't see any other sign of physiological or behavioral changes in these mice. So these are the results as tabulated. We have the control group with a consistent increase in total tumor volume and tumor incidence. The cisplatin did not significantly decrease the volume and the tumor incidence, whereas our group, which was exposed only to light, significantly decreased the tumor volume and the tumor incidence rate while preserving the survival of the mice. So we had 100% survival of the mice during the 8 weeks of treatment. So how we think this is working out, we have measured a lot of pieces from this scheme, but in brief, visible light, more specifically blue light, is capable of interacting with biological target. We hypothesize that it is the cytochrome C oxidase due to literature reports. Electron leakage leads to the production of reactive oxygen species. This was measured under our controlled light irradiation. It's important to note that our light irradiation here changes significantly from our light irradiation in photodynamic therapy and photoactivatable chemotherapy, since it is much more prolonged and much more intense. So here we are using the energy of light itself to trigger the photobiological processes. So the control of the light flux and the energy of light is very important when it comes to which therapy we are looking at. Upon the production of reactive oxygen species, we found out that we have the apoptotic pathways triggered, both the intrinsic and the extrinsic pathways were found to be involved in leading to cancer cell death. Now, what we have shown here is that a commercially available high-intensity white light can be used as a phototherapy against chemically induced skin carcinogenesis model. A significant anti-cancer effect was recorded upon short exposure. And here, when we say short exposure, it's 90 minutes of time on the dorsal side of the shaved mice. And the potential phototoxic mechanisms involved induced apoptotic pathways, and they are partly triggered by ROS production. And we also have found significant DNA damages in tissues and cancer cells that were exposed to light. Our results encourages the translation of this drug-free phototherapy into the clinical setting. So by that, I would like to thank Dr. Castellano from North Carolina State University for hosting me as a visiting scholar in his lab, allowing me to access instrumentation, specifically ultra-fast spectroscopy instrumentation that are multi-million dollars facilities. I would like to thank Dr. Basir from Balamand University for collaboration on the crystal structure elucidation. I would like to thank from LEU, Stephanie Mhanna, Najwa Mansour, and Hasib Aoudi for generating a lot of the data that I have talked about today and help with analysis. I would like to thank Dr. Wax for collaboration on computational modeling of ruthenium complexes, Dr. Khalil for continuous collaboration on several projects, whether in the environmental application or in cancer applications, specifically the copper project, Dr. Tino Daher for continuous collaboration on the ruthenium projects and co-supervising several students in his lab who worked on elucidating the biological mechanisms, Dr. Sibai and Dr. Abi Habib for the collaboration on the biology part, specifically the copper and some of the early investigations on ruthenium drugs. I would like to thank talented LAU students, specifically undergraduate students who helped me in the copper projects. We have also graduate students who helped in generating data on the ruthenium project. By that, I would like to thank also the granting agencies, CNRS, GSR funding, and early school funding when the school had money to spare for research. And I would like to thank you all for your attendance, and I'm happy to welcome any question you might have.
Thank you very much, Dr. Khnayzer. Really fascinating and very informative lecture. It makes me nostalgic of the days I used to do a little bit of science, but not at this level. So if you don't mind, I'm going to ask Dr. Hermanani to manage the questions because my internet connection is playing up and I don't want to ruin this amazing event. I can already see Dr. Gergis's hand is raised. So I will invite Dr. Gergis to speak, and then following from that, Dr. Hermanani will continue the questions. Welcome, Dr. Gergis. Nice to see you.
You are on mute.
Can you hear me now?
Yeah.
We can now, go ahead.
Okay, thank you. Thank you for organizing this wonderful event, and thank you, Rony, for a great talk. I personally really very much enjoy the early characterization of novel bioactive compounds, whether they be organic or inorganic. So I personally enjoyed your talk very much. I have some questions that are kind of very in broad range about the idea of photochemotherapy. I think a central challenge in cancer therapy is knowing sometimes if some residual cells, or what we frequently call the loners, that are a bit away from the main tumor but still in the vicinity of the organ, as those might be very dangerous ones that can be missed in surgery when surgeons try to remove the bulk of the tumor. And so I'm trying to interface this with what you're saying about the compounds. And how do you see in the long term the idea of how assuming your drugs are utilized in a clinical setting on patients, how one would be able to identify where some of the cells are? Because with biomedical imaging, I mean, we can see big tumor masses on the order of cubic millimeters, but we can't really see the individual cells, and some of them might be quite dangerous. How do you see this, kind of playing out on the long term?
Yes, thank you, Dan, for the comment and the wonderful question, because this is exactly what is the next basically steps in our research. What we are trying to think about is how to deliver these photoactivatable drugs specifically to cancer cells and not to others. So what we are trying to do now is trying to see if there are any receptors of interest where we can append our molecules and synthesize new ones that would bind to biological targets and be delivered specifically to the places where we want them to be delivered. So to that end, a big part of my research now is focused on developing folate receptor alpha targets for ovarian cancer. And folate receptor, as you know, is upregulated in various types of cancers, including ovarian cancer. So our aim is to try to do targeted drug delivery followed by light irradiation. The light can go into the body, to the ovaries, either through endoscopy or other technology. So this is really beyond the scope of my talk, but there are technological advances, whether in imaging or in therapy, that are basically supporting this technology. So I would like to draw your attention, Dan, that currently a lot of chromophores are being delivered to visualize these cells that you talked about, that typically these surgeons are missing. And my research was inspired by a publication in Nature Chemistry that highlights a simple tuning of folic acid to couple it with a simple dye and aid the surgeons in their surgery. So they used it for imaging purposes. So I thought if they could deliver the drug and visualize these, let's call them missed cells, why can't we do the same, but instead of delivering an imaging agent, deliver a photoactivatable drug. So we embarked on this years ago. We had so many challenges because these molecules are charged in nature and they like to bind to different targets by themselves, but we're still working on it and we have very promising results, but they are very preliminary in that perspective.
Can I ask a follow-up question if I can get away with it?
We need to ask.
Of course, go ahead.
Thank you. So the light thing that in your third part that you showed about so it made me think a little bit about your first part of the lecture when we were talking about the copper complex. So did you see an effect of light alone when you use the complexes? And the kind of a mini question in that same part as well. You mentioned you showed some data showing that the copper complex can bind to DNA. So I'm assuming that idea, along with the accumulation selectively in tumor cells, kind of is the edge of the sword in the sense for making this drug potent for killing tumor cells. But I think at some point you would also have to look at, so mesenchymal stem cells are still stem cells and they are rapidly dividing, but there are other stem cells and mature cells in the body that are rapidly proliferating that they're usually targeted by chemotherapies, and I think the rapid proliferation enhances the accumulation of a drug that binds to DNA, because if you have a cell mass that produces more DNA, then you have more binding sites, and that would perhaps kind of a feedback towards the accumulation of the drug. So the idea would be to test similar cells. I know melanocytes are quite cool for this, especially that you're using UV light, which is low penetrating. But the idea would be to use something more related to melanocytes, perhaps like normal epithelial cells or non-tumorous melanocytes. Have you thought about that? And I can imagine it could be quite hard to get these cells in the country.
Yes, actually, this is a reviewer's question. We have one paper under review, and the reviewer asked us to extrapolate on other kind of normal cells. But
I can assure you it's not me.
Yeah, okay, it's okay. So basically, our answer is that we have done it in vivo. So we have responded that we have done it in vivo, and basically we have, some of the studies related to that project were done also in vivo to check for selectivity, and we see no really cytotoxic effects on healthy tissues. We did all kind of studies, and we saw that the drug is being basically kicked out from the body with no obvious side effects on healthy tissues, while it really helps in mitigating the problems and the tumor volume. So, and of course, pathology was done and all of this, but I didn't have time to cover all of these different aspects of the project. My answer to you is that I wish we can grow these cells in Lebanon, and if you can, please let me know so that we collaborate, because always we have this kind of dilemma. Is it enough to do it on mesenchymal stem cells, or we need more controls to our experiments?
I can help you out on the breast cancer side, because we have normal human and tumor from human as well, from the same patient. So if you ever want to try it out, I'd be happy to help.
This is excellent. Thank you.
Thank you, Dan. Thank you, Rony, for the intriguing technical discussion. Now, on the other spectrum, we have a question from Dr. Behmardi, and that defines, in a way, all our questions, and that is, what does blue light mean? How do you probably derive it? And he also has a follow-up question in terms of whether natural light could help also in terms of cancer remedy.
Yes. So, Dr. Behmardi, your questions are actually very, very, very interesting to us as scientists. So first, as a definition for blue light, it's a light or photons that have a specific energy that coincides with a wavelength of light that is around 450 nanometer plus minus 20 nanometers. So it's really the energy of the photons that are hitting the biological target. So blue means that the color is blue, so you see the blue light, but also it means that the energy correlates with a certain wavelength of light that is around 450 nanometers. So I showed you how different wavelengths of light, different energy of light can interact differently with biological targets, or even with photodynamically active molecules, depending on their absorption spectrum. Now, regarding the effect of natural light, this is a philosophical question and a scientific question, because natural light throughout history was used as both beneficial to our human body, but also some aspect of the natural light or the sunlight can be harmful. For example, the UV part of the light is harmful. So I had similar question to see whether the harmful effect of UV light can be mitigated by the presence of blue light. It is a fundamental question. We don't have the answer for it now, but currently we are developing a series of experiments in vivo to answer this, and I hope I can share with you the results soon. If the blue light help in protecting the skin from developing cancer, that is a characteristic of UV light. So UV light is known to be a carcinogen or a cancer-promoting radiation. Now, the answer to the question whether blue light offers a protective effect is not known yet. But in general, natural light does have a lot of positive impacts on tissues. And I highlighted a few of the physiological functions of natural light, including bactericidal effects and stimulating effects on tissues.
Thank you, Rony. So next question, we have Dr. Khalil. Chris, go ahead.
Rony, thank you for this outstanding presentation. I really enjoyed it. It's really great work that you've done. I have a couple of inquiries. I think maybe these have been also flagged by the reviewers. You mentioned using mice. Have you tried different animal models, like, for example, rats or baboons, and see whether you see a similar correlation in different animal models? The other thing I have, which is of concern to me at least, is the fact that you're using only one cell type, which are the melanocytes. While in the skin, you have other important cell types like the keratinocytes, and perhaps the most important one in the immune reaction are Langerhans cells. So have you ever considered using these type of cells? And building on what you've said today, I had actually two ideas for future projects. One of them is using human skin from patients undergoing surgery for cancers. So maybe if you can get them and expose them to your compounds and see whether they are efficient in reducing the cancers in these human skin explants, we call them. And the other idea, which I found useful too, maybe you can shed some more light on it. I think the exposure time to get some impacts is quite long, because you're having 2 doses of exposure, I think, over more than 1 week of exposure. So maybe a good idea, maybe for future research project, there's a group in Queensland that used a compound that they have isolated from wild berries growing in Queensland forests. And this compound, from the seeds of the berries, they got this compound known as EBC46. And this compound is quite potent. If you inject it into the area where you have a cancer, you start seeing blue color or bruising within 5 minutes of injecting it. Within 24 hours, the tumor will turn black. And within, I think, 1.5 weeks, the tumor will just drop off and disappear. So maybe it's a good idea if we get this compound, maybe couple it with your photoactivated compound, and maybe that will also be very helpful in attacking different types of cancers. Especially this compound have been tested on melanomas, on skin cancer, on head cancer, on neck cancer. Again, this is an idea, but I think you've done a very good work, very, very professional, and good luck.
Thanks, Chris.
But Dr. Khalil, you have asked many questions. I hope I can remember them. I was not taking notes. So regarding the multiple studies, yes, of course, we in all our studies, we include many cell lines to start with. Screening, call it screening. So in ruthenium prodrugs, we did maybe 6 or 7 cell lines, but I just showed a representative data on those. In others, the minimum we test is 2 to 3 cell lines per compound. And of course, if we find certain potential activity, we extrapolate into other cells. Now, regarding the in vivo models, I really hope with the new animal facility, we can do more tumor basically or cancer models. And our collaborators in the biology program are very excited for the capacity of the new animal facility. And I hope just to basically be able to run these experiments as soon as possible and have money to feed the animals that you are mentioning, because as I know from Dean Jnaynati, feeding the animals is also something that we have to factor in terms of major costs. So yeah, definitely this is on our mind. We hope we will be able soon to develop new cancer models. Now, regarding ex vivo, kind of like removing the tumors and testing it outside the body, this is an extremely interesting approach, and I think we have talked about this before. It is limited by maybe the viability of these cells and how much we can do in terms of showing long-term activity. But yes, it is very interesting to look at those immediate response from extracted tissues. And actually, we are collaborating with a brilliant dermatologist in Rizq Hospital who is now providing us with basically more information on how we can move some of our studies into the clinical settings. Of course, this includes a lot of IRB approvals and other ethical approvals that we need to worry about. So I hope I answered most of your questions, Chris.
You did. Thanks.
Okay. Thank you, Rony. Thank you, Chris. We have a question from Dr. Nicholas. Go ahead.
Hi, Rony. Amazing talk. Thank you. I just have one small question. I was fascinated by the blue light experiment. I did not know about that. So my question there is, was the light like focalized? Did you try to play on the intensity to shorten the exposure time? And did you try at some point, I don't know if we have them, but like pulsed lasers, because these could change completely how the heat would dissipate in the cells and maybe would give totally different results. And then the second question, if we are able, or not question, maybe suggestion, if we are able to measure the heat capacity of those tissues, then we can have models where we can actually estimate how much they would heat up upon an exposure of light with a certain wavelength and a certain intensity for a certain time. So we can manipulate all of these parameters to reduce the experiments, like just target what we think would work.
Yes, thank you, Rana, for the question. Actually, we never tried the other type of irradiation. So we were focused on LED lights because they were cheap and available in our lab. So it would be very nice to extrapolate to laser since it is focused and we can basically have much higher photon flux in the laser experiments. Regarding heat, we didn't look into the microscopic properties of heat production. Rather, we have looked at the overall temperature of the skin and the tissue, and we saw using infrared measurements that we do not have any change in the temperature profile, which is expected for blue light. As you go more and more to the infrared, heat can be generated by from water, interaction with water and other molecules that can basically dissipate the energy in terms of vibrational relaxation. But it would be nice to simulate this, and maybe at the microscopic level, we are producing some kind of side thermal process that is helping in the overall therapy.
Thank you. Super talk. I really enjoyed it. Thank you again.
Thank you, Rana. Thank you, Rony, again. Any other questions? Okay, so before we go, there's a follow-up question from Dr. Behmardi on the chat box. So I don't know, Rony, if you've seen it. So Dr. Behmardi is asking whether people, I mean, based on your experiments, if they can somehow, I assume, can extrapolate and then somehow they can find a cure in the same way they have done with COVID and vaccine. I do think there are some vaccines for cancer. I don't know if you can elaborate on this, Rony.
Yes, actually, this issue of vaccination as a basically precaution, let's say, precautious measure for against cancer is not widely used and discovered, but there were certain type of cancers that are amenable to such basically vaccination. And last week, if I recall well, there was a group of researchers who have found a promising results on a specific type of cancer. I cannot recall, maybe Dan can recall more on how the use of a specific vaccination for that type of cancer. You cannot have one solution fits all. There's still the targeted therapy in certain kind of cancers, other therapy in other types of cancer. In addition to all of this, when we think about vaccination, we think about the immune system. So we are promoting basically the immune system to identify and target cancer cells. However, cancer cells are very smart because they are part of our cells. They are not basically coming from the environment, such as the viruses and bacteria. And that's why certain cancer cells, they can evade our immune response. And that's why they are trying to train the immune system to recognize cancer cells. And these are all ongoing studies. And I know I have a friend in the US, his kid was diagnosed with leukemia, and now they are experimenting on the use of this type of training the immune system of the kid to recognize cancer cells. So it's still in the experimentation phase.
Thank you, Rony. Any other comments or questions? Okay, so since we're almost done, I think for next week, before we go, I'd like to announce our next talk. Our next talk will be by Gigi Ghaddar. She's actually a candidate in the faculty of information at University of Toronto, and she'll be our speaker next week. So please tune in next week for Ms. Ghaddar, and looking forward to see you then. And thank you, Rony, again for this amazing talk. Thank you, everyone, for attending. Have a wonderful day, and we'll see you next week.
Thank you, Haidar, and thank you all for attending. Bye-bye.